Good morning, good afternoon, good evening. This is Jin-Sun Yu, CEO of Pharmapsin, a clinical stage biotech, publicly trading at Kosdaq stock market in Korea. We are focusing on development of next generation immuno-oncology drug for cancer patients.
So I'm going to start with executive summary. So what we actually are looking for here. Our top priority is equity investment. We are looking for strategy investment or financial investment or combined. We are looking for opportunity of licensing out for our clinical stage Olynvasmab and preclinical stage PMC 309 and PMC 43. Alternatively, we are also looking for co-development partners for Olynvasmab, PMC 309, and PMC 43. So the take-home message of our clinical stage Olynvasmab, it is anti-VEGFR2 neutralizing antibody. It's a fully human. Unique value is it has a cross-viscous reactivity, so it binds murine VEGFR2 up to human. So you can perform all kind of in vivo studies in animal model system, although it's an angiogenesis inhibitor. We did our Olynvasmab recurrent GBM phase 2 in Australia. We got 25% DCR, and more than 40% of patients have brain edema reliefs. And it always shows clear safety profile. We work with Merck together for combo trial with Olynvasmab and Keytruda for metastatic triple-negative breast cancer. And we presented at San Antonio Breast Cancer Symposium last year. And key message was clear safety profile from this combo regimen. And we have a 50% objective response rate, which is quite high in metastatic triple-negative breast cancer, and 67% disease control rate, although it's a small size of studies. So Pharmapsin and Merck agreed to initiate global phase 2 study for metastatic triple-negative breast cancer patients. The next key message from PMC 309 is, again, it's a preclinical stage and it targets human VISTA. So PMC 309 is anti-VISTA antibody antagonizing. It blocks VISTA-VISTA interaction and VISTA-V6-3 interaction, and also VISTA-PSGL1 interaction in various pH environment. Actually, VISTA-PSGL1 interact in low pH, which represented the tumor microenvironment pH levels. PM309 also increased primary T cell activation, but it inhibits Treg proliferation and activation. PMC309 has a stable PK. The half-life is more than 4 days, and it has shown clear safety profile in monkey, although it's a preliminary PK and tox studies. We are heading to GLP PK and GLP tox studies soon. Interestingly, PMC309 will change cold tumor to hot tumor in syngeneic mouse model system in knock-in mouse, and also has shown synergistic anti-cancer biology with anti-PD-1 antibody combined in mouse model system. The last asset we would like to expose is PMC43. it's a human Tie2 activating Tie2 downstream pathway. What we get, it renormalize pathological leaky vessels. It has cross-viscosular activity. It binds and activates murine Tie2, but monkey and human. Interestingly, it doesn't bind VEGFR2, but it inhibits the VEGF-VEGFR2 pathway simultaneously. And also, interesting observation we made is it reduced the Tie1 copy numbers. And it has shown significant anti-tumor synergy effects with anti-PD-1 antibody in mouse model cyst. This slide shows the table with our pipelines and development status. So come back to olinbasimab. Since it blocks VEGF-VEGFR2 pathway, we believe it's an anti-tumor angiogenesis inhibitor, but also we observe transient tumor vessel normalization and also immunomodulator in tumor microenvironment. Its current clinical trials, we are running phase 2 mono for Avastin relapsed refractory recurrent GBM patients in USA and Australia. We are running phase 1b combo with Keytruda for metastatic triple-negative breast cancer and recurrent GBM patients. Our development strategy, Pembro combo global phase 2 trial for mTNBC is executed with Merck. And we are also planning for various mono and combo with various modalities in different tumor types. Its competitiveness is, first of all, clear safety profile. And not all cancer, but certain cancer type, olinbasimab already show superior efficacy biology. So this slide shows olinbasimab clinical development status, and Famapsin is mainly focusing on Pembro combo for metastatic triple-negative breast cancer. Although we are still considering to do recurrent GBM phase 2b pivotal studies. But also, as I mentioned, we are looking for other modality combo and other type of cancers by olinbasimab monotherapy too. This is kind of a summary we presented at San Antonio Breast Cancer Symposium, showing that in the cohort 60 mg/kg weekly dose of olinbasimab plus Keytruda flat dose deliver 50% ORR, 67% DCR, and one patient with PR had the CR in the target lesions. There are some adverse effects, mostly come from Pembro, like hypertension and pulmonary embolism. And this is the spider plot. As I mentioned, 60 mg/kg provide better efficacy data. This is competitive landscape of olinbasimab. As you know, Avastin from Genentech Roche is already marketed, and biosimilars like Mvasi, Jiravev is on the market, and also VEGFR2 neutralizing antibody from Eli Lilly called Cyremza is already marketed since 2014 for stomach cancer and various tumor types. And Geltrap from Regeneron is also on the market. However, they all have a common side effects like hypertension, GI perforation, hemorrhage, and proteinuria. Uniquely, olinbacimab, we never observed such a side effects. What we observe is reversible capillary hemangioma on the skin, mostly neck and chest area, mostly grade 1 and sometimes grade 2, but no grade 3 or beyond. So very safe. I'm going to shift the gear to PMC309, anti-VISTA antibody, kind of next generation immune checkpoint blockade from PD-1, PD-L1, and CTLA-4. So it's kind of negative regulators. VISTA highly expressed on MDSC, and aggressive tumor microenvironment has high copy number of MDSC to maintain quite immunosuppressive environment. And VISTA signaling plays pivotal role to proliferate regulatory T cells and activation of regulatory T cells. So PMC309 block the VISTA biology and inhibit the regulatory T cells, but boosting effector T cells. We are heading to IND early 2022, and we are initiating phase 1 dose-limiting toxicity studies as a mono, but also combo with IO or other modalities. It's first in class. There are a couple players already in phase 1s, but we believe our PMC309 has a unique value compared to other VISTA antibodies from competitors. Like other competitors, PMC309 very specifically bind VISTA, but it binds only human VISTA. And in low pH, our competitor bind just the CHOK1 MOC cell too. So our antibody has a very specific binding property and affinity as a nanomolar range and subnanomolar range. PMC309 also binds to human myeloid-derived cells, but not to T cell, as you can see. CD4, CD8, Treg, no binding, just NK cell binding, and all MDSC-derived cells, PMC309 bind very well. And we also made an assay system by using VISTA-FC, recombinant protein, which show that it suppresses both T cell activation and proliferation very well. And PMC309 also inhibit broader spectrum of VISTA interaction, but also VISTA V6-3 interaction and VISTA PSGL1 interactions. And we believe that this specific interference will be beneficial for cancer patients. PMC309 enhance interferon gamma secretion by human PBMC very well, as you can see on the slide. And in this assay, we also has shown that PMC309 enhance T cell activity very well. We cannot compare with anti-CD20 antibody, but still our IgG1 based PMC309 has some mild ADCC effect compared to IgG4 derived PMC309. And we also has shown that PMC309 has a phagocytic effect in VISTA expressing cells. So PMC309 bind MDSC, and this complex were targeted by macrophage by their phagocytic biology very well. In that way, PMC309 can reduce the number of MDSC in tumor microenvironment. Interestingly, also PMC309 synergized pro-inflammatory effect with anti-PD-1 antibody, meaning that PMC309 has a biology to change cold tumor to hot tumor, so that anti-PD-1 antibody or other immune checkpoint blockade, which has less active in cold tumor, has a better performing biology in the presence of PMC309. And PMC309 PK profile in sea monkey, we have shown about 4 days half-life, which is not bad. And it is still a preliminary study, so we're gonna get the complete GLP PK data soon. And also PMC309 has a very clear safety profile in monkey. Again, it's still preliminary data. We're gonna update with GLP tox data once we have everything. Again, several players have anti-VISTA antibody. Most advanced one is Curis VISTA antibody, which was initiated phase 1 by J&J a couple years ago, and there are a couple others who are preparing for their IND2, like Famapsin. So the last molecule I would like to introduce is PMC43. That's an anti-Tie2 antibody, bind to Tie2 and activate Tie2 downstream signaling. True value is it normalize pathological leaky vessels. And it can be applied various disease indications, and it's the first in class asset. There is no Tie2 antibody which activate Tie2 downstream signaling so far. And this is kind of collection of application we can try. We are eager to show whether PMC43 has some synergistic effect with anti-PD-1 antibody. And we have shown that there's a significant synergy effect from combination with anti-PD-1 antibody. And it improved the T-cell infiltration to TME and etc. Not only in oncology, but we are also exploring in ophthalmology area. And we got from murine, but also monkey CMB models that PMC43 has excellent biology to repairing this pathological vessel damaged by laser treatment. So this is competitive landscape. Again, PMC43 is the only Tie2 directly binding activator so far. So it is really interesting and exciting competition, I would say. So thank you very much for your attention, and I would be happy to answer any questions if you have. Thank you.